So let us discuss about HSV1 virus and HSV2 virus. So it is icosahedral DNA double-stranded DNA virus, and this causes the oral or ocular infections, and this causes the genital infections. HSV1 and 2 both can be distinguished. You can see this in our another video on going on to our channel, and I will link the video below if possible. Now let us talk about the pathogenesis. So it first infects, suppose it is infecting our eye, so it will infect the eye first, and then it will form vesicle. Vesicle is formed, which is spread to lymph nodes, and through lymph nodes it causes lymphadenitis, that is the swelling of the lymph nodes. So after causing lymphadenitis, it flows retrogradely in axon. Retrograde is opposite flow. The flow into the axon is from body to the dendrite. However, this virus or this infection flows retrogradely from dendrite to axon, and is most commonly in, it is settled within the neurons in ganglia. So HSV1 is in trigeminal, and HSV2 is in sacral root ganglia. So this also is one differentiating features among these two. Now next, what can we do for the laboratory diagnosis? For the lab diagnosis, we can do following. First is, what will you take as specimen? So for specimen, you can take fluid swab or the skin swab, saliva, or disinfecting cornea. So you can take the corneal swab. Now for the direct examination, you can take the scrapings from the vesicles, and toluidine dye, toluidine blue dye is used for staining it. In that you will see multinucleated giant cells, and these are stained as ground glass chromatin. And this is also called as the Zang cells. So what can you do for early diagnosis? For the early diagnosis of encephalitis, you can do brain biopsy, which can tell you the result very fast. Now next you will do tissue culture. In all the virus, you always do tissue culture or culture in cell lines or in animal inoculation. So Hep2 cells is mostly used in all of the viruses. E is capital in this, and I had told you chorioallantoic membrane in the other video, so you can grow it on chorioallantoic membrane. One thing is more, HSV virus will produce syncytium, which HSV1 virus will not do. For the serological test, you can do a check for antibodies, IgM antibodies. You can do ELISA, RIA, and PCR and complement fixation test also. So this is what you should write if you get short note on herpes simplex virus 1 or 2. Thanks for watching.